Welcome to part 3. In this part, we're going to look at the transport of gases through the blood. And I'm sorry, but it is going to contain a bit of chemistry. We're then going to go on and look at the regulation of blood gases and how that has an impact on respiratory rate and depth. As we know, most of oxygen is carried around by hemoglobin within the erythrocytes or red blood cells, but that is only 98.5%. 1.5% of oxygen is dissolved in the plasma and cytosol. Each erythrocyte has around 250 million hemoglobin metalloproteins. Each of these metalloproteins, they're called metallo because they have this, has 4 iron Fe++ containing heme groups. So within each metalloprotein there are 4 heme groups. And within each erythrocyte there are 250 million hemoglobin metalloproteins. Each of these ions on the heme groups binds 1 molecule of oxygen, and that's at full saturation. The binding of oxygen to hemoglobin to form oxyhemoglobin is called oxygen saturation, or SO2. Saturation is determined by the partial pressure of oxygen within the blood, but also the affinity of the hemoglobin for oxygen. So, for example, it decreases during exercise so that more oxygen is released from hemoglobin to diffuse into the cells. We'll talk more about saturation in a sec. So let's look at that structure again. So here's a hemoglobin, or a hemoglobin metalloprotein. You notice you've got 4 different chains. 2 of them are alpha, 2 of them are beta. But that's not really that significant for us at this stage. What you will notice is that there are also a heme group per chain. Within each of those heme groups, we have an ion of iron. That's what oxygen binds to. The 2 beta and 2 alpha chains therefore have 1 Fe++ containing heme group each. And remember, there are 250 million hemoglobin metalloproteins in 1 erythrocyte. Each Fe++ can bind 1 oxygen molecule, and this is dependent upon saturation. And you'll notice here that oxygen is a molecule, and of course it's labelled O2, the 2 implying that there are 2 oxygens, making it a molecule. Talking about saturation, you'll come across probably in texts about the oxygen hemoglobin dissociation curve. We're not going to go into that into too much detail, but basically this curve, it's a graph. It plots the saturation of hemoglobin against partial pressure in the blood. It's determined by this affinity, the affinity that each Fe++ has for oxygen to bind it. Generally, it's a steep curve at the start, and this is because the binding of the first O2 molecule to a hemoglobin is harder. Once you bind 1 O2 to the hemoglobin metalloprotein, the hemoglobin changes shape, and this facilitates the binding of 3 other oxygen molecules to the 3 other heme groups. Generally, you'll see that the curve levels out as well. It plateaus at the top, and this is because the hemoglobin has become saturated with oxygen and no more combined. So what about carbon dioxide? Well, 20% of it is bound to hemoglobin, but not to that heme group. We'll see why you have some in hemoglobin in a minute. 70%, so the most of it, is converted within erythrocytes into bicarbonate, HCO3-, for transport in the plasma. 7 to 10% is then dissolved in the plasma as CO2. So let's look at that conversion. So during internal respiration, the cells, after cellular respiration, release CO2 that diffuses out following its pressure gradient into the blood. When it leaves the cells, first of all, it diffuses into the cytosol of the erythrocytes. So it enters the erythrocytes. Inside the erythrocytes, there's a high concentration of an enzyme called carbonic anhydrase, or Ca. Carbonic anhydrase then catalyzes a reaction between carbon dioxide and water, and this forms carbonic acid, H2CO3. Carbonic acid quickly dissociates into bicarbonate, HCO3-, and our old friend protons, H+. The bicarbonate then diffuses out of the erythrocyte back into the plasma, and it does this in exchange for chlorine because we need to balance those charges, both being anions. So here's that equation. So we start with carbon dioxide and water, and through the action of carbonic anhydrase, we get carbonic acid. Carbonic acid quickly dissociates into bicarbonate and protons. And I've given you the equation in words and in chemical symbols. One thing to notice here is that I've got a little double arrow between it. This is because this equation is reversible. This reaction can work both ways. So if you think about that, why do you think we need to do that? Well, if you think about it, when of course the bicarbonate gets up to the lungs, we can't breathe out bicarbonate. So it has to be reconverted back to carbon dioxide for us to exhale it. So we've just seen that carbon dioxide is converted to bicarbonate and protons on release from the cells during internal respiration. After transport as bicarbonate, in preparation for external respiration, the bicarbonate returns to the erythrocytes and is converted back to CO2 via the reverse of the previous chemical reaction. Okay, so let's look at the transport of those gases. So up here in internal respiration, we have our alveolus. Down here in internal, we have our cell. In between, we have our systemic circulation. So let's have a great big circle. Again, there we go. Need lots of room in this circle. So we've got nice big fat vessels so that I can draw in. Okay, so in terms of oxygen and transport, it's fairly straightforward. Oxygen, we're not going to talk about the partial pressures this time, but bear in mind that everything, of course, is driven by the partial pressures. Oxygen moves into the blood, where it finds its erythrocytes. So there's our nice erythrocytes. These erythrocytes, remember, contain these hemoglobin metalloproteins that are full of iron, Fe++, to which oxygen is going to bind. So our erythrocyte has hemoglobin. When oxygen gets into our erythrocyte, we get oxyhemoglobin. So we have HB for hemoglobin, O2 for oxygen. And that, of course, is dependent upon saturation. Saturation. And several things affect saturation. Remember, we're talking SO2 being the symbol for saturation. Okay, so we've got our little erythrocyte, and bound in the hemoglobin metalloproteins, potentially 4 oxygen molecules per hemoglobin, depending on saturation. That's going to travel round in the circulation just like that. When we get to where it's needed, here's our erythrocyte, oxygen is going to disassociate from the hemoglobin so that it can diffuse into the cell. So oxygen disassociates from hemoglobin. So oxygen's fairly straightforward. Carbon dioxide, of course, is where we start to get into our chemical equations. So in terms of carbon dioxide, in order to diffuse out of the cell, it diffuses out as carbon dioxide. So here we have CO2 diffusing out of the cell. To be transported, it needs to be converted to bicarbonate. For that, it needs erythrocytes, not the hemoglobin component. So we have an erythrocyte. Erythrocyte, so carbon dioxide, moves into erythrocytes. This is in internal respiration. Within erythrocytes, we have a lot of carbonic anhydrase, and that's what we need there. Carbonic anhydrase. Carbonic anhydrase hydrates CO2. So if it hydrates it, we're adding water. So we have CO2 plus H2O, which then gives us carbonic acid, H2CO3. And you can see, if you look at it, where all those H's and the C and the O's have come from here. But of course it doesn't stay as carbonic acid, it quickly disassociates. We're still within the erythrocyte here, this is all happening in here. Carbonic anhydrase is causing this to happen. When we get to carbonic acid quickly disassociates, and it disassociates into our bicarbonate. So we end up with HCO3 minus. Yes? And some protons. Most of those protons actually stay within the erythrocyte, but some do come out. But primarily, we're talking about the bicarbonate coming out to be transported in the blood. Yeah? Okay, so that's fine. We've got our bicarbonate in the blood, in the plasma, gets back up to the alveolus, to the lungs. But of course, we can't diffuse out bicarbonate. It's got to go back to being carbon dioxide. And that's why this equation has to be reversible. To do this, again, it needs carbonic anhydrase. And where we find that is erythrocytes. So it goes back into an erythrocyte. Inside the erythrocyte, we have carbonic anhydrase. What that does this time is rather than hydrating, it's dehydrating. So we have, that's my green, HCO3, and that, and our protons, and H+, that, if we reverse this back again, what was our step before? It was carbonic acid. That's converted to our carbonic acid, H2CO3. Carbonic acid, again, doesn't like being carbonic acid, so it quickly dissociates, and we get CO2 and water. CO2 diffuses out of the erythrocyte and into the alveolus. And that's how transport links up with external respiration and internal respiration. So how about acidosis and alkalosis? Well, if we have an increased CO2, we will naturally have an increase in bicarbonate and, more importantly, an increase in protons because they're produced at the time of the conversion to bicarbonate. Increased protons will give us a low blood pH, and this we know as respiratory acidosis. So approximately a pH of less than 7.4. Most cases of respiratory acidosis are more about an inability to eliminate CO2 rather than any sort of increase in production by the cells. Hyperventilation, on the other hand, can give us the reverse, alkalosis. In alkalosis, we have a low level of protons and a high pH. And you can see on the graph at the bottom, on the left-hand axis, we've got bicarbonate and consequently H+. On the bottom, we've got increasing pH. So, increased H+ gives us low pH and acidosis. Low H+ gives us high pH and alkalosis. So in terms of lung capacity, if you look to the middle of the graph and you'll see the wavy blue line within the two other lines and the green arrow, this is resting tidal volume. So this is normal quiet inspiration. And the volume is generally around about 500 mils. 150 mils of this, however, stays within the airways to keep them open. Above that, if you breathe in really, really hard, you get inspiratory reserve volume. And that gives you a better idea of lung capacity. Conversely, if you breathe out and force it right the way out, and completely shrink your lungs down as small as they'll go, you get expiratory reserve volume. The two of those, the two extremes, then give us the vital capacity. Total lung capacity includes that 150 mils that keeps the airways open. To do all this, we need to use accessory muscles. And this is where we start to see changes in the chest wall that indicate to us that someone is using their accessory muscles. Okay, so we've talked about acidosis and alkalosis. In acidosis, we have a low pH. We have a low pH because we have a high level of bicarbonate and protons, because we have high CO2. What we need to do in this instance is to increase respiration rate. So by doing that, we're blowing off CO2, getting rid of more CO2. That's going to bring down CO2, bring down H+, and then increase our pH back to normal levels. And the opposite goes in alkalosis. In this case, we need to reduce respiration rate. So the question is, how do we do all this? Well, carbon dioxide is the main driver for increasing or decreasing respiratory rate and depth in healthy people. However, the problem is we need to know that these levels are up or down. So we need some mechanism for monitoring our own blood gases, an endogenous system. This is primarily the role of the respiratory chemoreceptors. So let's get an overview of these chemoreceptors. They come in two classifications according to their location or general location. First, we have the central chemoreceptors. These are found in the medulla, very close to the respiratory centres. They monitor H+ and therefore CO2 levels, but they monitor it in the CSF, the cerebrospinal fluid. It's a minute-by-minute regulation of CO2. However, the central chemoreceptors do not monitor oxygen. The peripheral chemoreceptors, these are found in the arch of the aorta and in the carotid arteries, in the carotid sinuses, so very near to the cardiac baroreceptors that monitor blood pressure. The respiratory chemoreceptors are known as aortic and carotid bodies, obviously depending on where they are. These monitor H+ and therefore CO2 and pH, but they monitor it in blood, arterial blood. They tend to only respond to large drops in oxygen, but they do monitor hypoxia. So if we have a drop down to about 60 millimeters of mercury in arterial blood from 100, bear in mind, then they tend to start responding then. Hence, the respiratory regulation is primarily driven by CO2. So let's talk about the central chemoreceptors first. Remember, these are monitoring H+ in the CSF. They're found in the medulla. So H+ can't itself cross the blood-brain barrier, but CO2 can, and this diffuses into the CSF from the blood. In the CSF, CO2 is then converted by the same process as we had in the blood. In other words, CO2 and water become carbonic acid by the reaction of carbonic anhydrase. Carbonic acid then quickly dissociates to become bicarbonate and protons. So this all happens within the CSF. The protons that are released from this then bind to the chemoreceptors. Remember, chemoreceptors are just that. They're receptors that bind chemicals, in this case H+. If enough of these chemoreceptors are bound by H+, we get an action potential that is sent via little interneurons to the central pattern generator in the medullary respiratory center. This increases the firing of the inspiratory cells and delays the firing of the expiratory cells. Therefore, we get increased respiratory rate and depth. This, of course, then helps to blow off the excess CO2, as we said. Which brings us to these peripheral chemoreceptors. And you'll see in the diagram the locations. We've got the aortic bodies, remember, and the carotid bodies. The peripheral chemoreceptors also monitor H+, and therefore CO2 levels, but they do it in the blood. They're not monitoring CSF, they're monitoring the levels in the blood. The mechanism is thought to be similar to the central chemoreceptors, but it's faster because we don't have to have this conversion within the CSF. The afferent autonomic neurons that come from these, so the action potentials created from these aortic and carotid bodies, are the vagus nerve from the aortic bodies and the glossopharyngeal nerve from the carotid bodies. So again, very similar to the baroreceptors. And remember, these are both cranial nerves too. However, unlike the central chemoreceptors, these peripheral chemoreceptors in both the aortic and the carotid bodies monitor PO2, oxygen levels, within the blood. They usually only respond to a significant hypoxia. So as I said before, we're talking about a PO2 of less than about 16 millimeters of mercury. So a drop of 40 from our usual arterial 100 millimeters of mercury. And this is generally inhibited by the reserve carried by hemoglobin. Hypoxia is detected in both the carotid and aortic bodies by these specialized cells known as glomus cells. Incidentally, in chronic lung disease, you'll remember I said before, in healthy people, PCO2 is the driver. However, in chronic lung disease, hypoxia becomes more of a driver of respiratory rate and depth. So how do glomus cells do it? Well, they have oxygen receptors on the extracellular surface of their plasma membranes. These receptors are then associated with potassium channels. If oxygen is low, there will be no oxygen bound to these receptors. When no oxygen is bound to the receptors, the potassium channels close. This stops potassium from effluxing following its chemical gradient. And this retention of potassium causes a local intracellular depolarization. This is enough to open voltage-gated calcium channels within the glomus cell plasma membrane. Hence we get an influx of calcium. The influx of calcium then interacts with the cytoskeleton, and you know what happens now. We get exocytosis. In this case, we get exocytosis of the neurotransmitter dopamine. Dopamine then creates an action potential. This action potential travels via the afferent vagus and glossopharyngeal nerves. Remember, we've got the vagus from the aortic arch, glossopharyngeal from the carotid bodies. The action potential travels up to the dorsal respiratory group and then is transmitted to the central pattern generator in the ventral respiratory group. The ventral respiratory group then increases rate and depth. Of course, if you increase the rate and depth, you're going to be taking on more oxygen. So more oxygen will be available and the levels will rise. Okay, so monitoring hypoxia. This is where our glomus cells come in, and they've got a really funky mechanism. So in the aortic and the carotid bodies, we have these glomerulus cells. So here's our nice glomerulus cell. The glomerulus cell is, if you like, it's a chemoreceptor. So because it's a receptor, of course, it's got to send its signals, and for that it needs a neuron. So below it we have a synaptic cleft, and we have a neuron. This neuron is going to go to the dorsal respiratory group. Because remember, the dorsal respiratory group takes all the information from the respiratory chemoreceptors. The nerve, if you remember, if we're talking about aortic bodies, what nerve are we talking about? Good old friend, the vagus nerve. If we're talking about the carotid bodies, we're talking about the glossopharyngeal nerve. Glossopharyngeal nerve. So depending where this glomerulus cell is, which set of peripheral chemoreceptors, will depend what neuron what nerve this neuron is traveling into the DRG. Okay, so let's get back to our glomerulus cell. The glomerulus cell, remember, we're monitoring oxygen in the blood. So our glomerulus cell is going to be in direct contact with the circulation. It has to be able to monitor oxygen. So here's our glomerulus cell. Okay, glomerulus cells have potassium channels. Now these are oxygen sensitive potassium channels. So they sit in contact with the circulation and they bind oxygen. So here's our oxygen bound. When oxygen is bound, the oxygen sensitive potassium channels remain open. However, in hypoxia, there is less likely to be oxygen, and it's less likely to be bound to that receptor. So when we have hypoxia, quite a significant hypoxia, so we're talking about below 60 millimeters of mercury in an artery, bear in mind that the usual is 100. So in significant hypoxia, there will be no oxygen bound. When there's no oxygen bound to this channel, it would do the reverse, and the potassium channel will close. I'm trouble with my pens. So let's close that off. So here we've got no O2 bound, i.e., we've got hypoxia. Okay, so we know potassium likes to move in and out of the cell quite happily, and that then determines resting membrane potential. If it closes, potassium is retained. So potassium can't leave the cell. So it builds up. A little bit builds up, and it gives us, because it's positive, a little area of local depolarization. Okay, That's nice, isn't it? But what we also have here is a voltage-gated calcium channel. There's its little voltage sensor. It's voltage gated, therefore it responds to depolarization when it reaches threshold. When the little voltage sensor reaches threshold, because we've retained this potassium, the calcium channel will open. There we go, we'll open that one. And of course, we get an influx of calcium. Here we go, we'll have an influx of calcium. Now, calcium is often responsible for exocytosis because what it does, it interacts with the cytoskeleton. All cells have that cytoskeleton, and it's made up, or part of it's made up, of contractile myofilaments. Calcium binds to the cytoskeleton. The cytoskeleton contracts, and when it contracts, what it does, it moves vesicles. Now, in this case, we have vesicles of dopamine. Dopamine is a neurotransmitter, and it's our neurotransmitter of choice in this circumstance. So the cytoskeleton moves these vesicles to the membrane, a presynaptic membrane, and we get exocytosis of dopamine into that synaptic cleft. Like most postsynaptic membranes, we have ligand-gated or chemically gated sodium channels. So here we go. Here's our little sodium channels. And dopamine binds to those ligand-gated channels. When that happens, our sodium channels open. And of course, as always, we get an influx of sodium. This is a neuron. When we get an influx of sodium, we are going to potentially get an action potential. So that sets off an action potential that travels along either our vagus nerve or our glossopharyngeal nerve to the dorsal respiratory group. At the dorsal respiratory group, it receives the action potential and it says, oh, we haven't got enough oxygen. It doesn't actually say that, but that's how the mechanism works. It knows now that there is hypoxia. If we have hypoxia, we need to breathe more. We need to increase respiratory rate and depth. So the DRG, dorsal respiratory group, communicates, probably via little interneurons, with our VRG. If you remember, we've got the pre-Botzinger complex, that's our central pattern generator. And what that does is increases respiratory rate and depth, so that we can take on board more oxygen and resolve that hypoxia. So there are other factors that affect rate and depth apart from these chemoreceptors that we've been talking about. We also have chemoreceptors within the muscles, and during exercise, so that we get more oxygen for muscle contraction and relaxation, these chemoreceptors will increase respiratory rate and depth to take more oxygen on board. Also, we have the limbic system, which is our emotional center, and speech patterns. Remember, we said we need to change for speaking. We need to change the rate and modulate it and change the regularity. And this is all done through the Pontine Respiratory Centre. Voluntary interruption, of course, we know you can hold your breath. But this is ultimately going to be limited by non-voluntary regulation, even in small children having a tantrum. The autonomic nervous system has an impact. Remember, the sympathetic nervous system upregulates everything, including respiratory rate. And conversely, the parasympathetic nervous system downregulates. We also have these J receptors. These are actually C fibers, and for a while no one knew what they did. But we have these J receptors, and they're found in the alveolar walls. And what they do is they promote rapid but shallow respiration during exercise so that we can take on more oxygen for exercise. They also happen to do it if you've got vascular congestion or pulmonary embolus as well. Aside from that, we have stretch receptors, and these stimulate something called the Hering-Brewer reflex. So we have these stretch receptors that basically stop us from breathing in too deeply. So it inhibits excessive inspiration and cuts it off. Apart from that, we've got things like cough receptors. If you think when you cough, you have to breathe in deeply and then let it all out. So the cough receptors do affect rate. We've got things like thyroid hormones. Thyroid hormones will increase the rate as well. So there's lots of other things that will alter respiratory rate and depth apart from the chemoreceptors. But the chemoreceptors are the main method of regulating respiration. Khan Academy has a very good resource on respiratory physiology. There are his usual drawings, and there's also some quizzes to help test your knowledge. So that was the final part of the respiratory lecture. As always, please make your own notes and your own diagrams, learn the content of all three parts, discuss it with others, with your study buddies, to try and help your memory and understanding. If you have any queries, there's any bits you don't understand, then please email me or ask me when you next see me.